Welcome to week 15 of Pharmacology for Vocational Nursing 1. This week we're going to cover three topics: the antiviral drugs, the antitubercular drugs, and the antifungal drugs. There'll be at least 3 audio lectures. We'll start with talking about the antivirals, the virus effect on the body, HIV, herpes, varicella, and the drugs used to treat them. Finally, we'll cover the nursing implications for the drugs. So let's get started. Let's do a quick review of the general principles of virology. You'll remember that viruses are much smaller organisms than the bacteria, and they're obligate intracellular parasites. So that means for viral replication, they can't replicate on their own. They have to attach to and enter the host cell. They're very simple structures, and they consist of a genome, a capsid, and an envelope. Now, mature viruses are called virion, and that is when they are complete and an infective form. They then live outside, or can live outside the host cell. They move outside the host cell. The genome is the inner core of the virion, and that's composed of either a single or double-stranded DNA or RNA, but not both. The viral capsid is this protein coat that surrounds and protects the genome, and it's needed in the fusion of the virion and the host cell. So the envelope then has a lipoprotein structure that contains viral antigens that are often chemically specific for various proteins on the host cell's membrane. So the envelope can also aid in the fusion process, but we need to remember that not all viruses have an envelope as part of their structure. So let's do a little bit more detail about how this process works. Just a quick review. We have step 1, where the virion attaches to the host cell in preparation for infecting the host. And then the viral genome passes through the plasma membrane, and at step 2, we've got this virus the virion inside the cytoplasm of the host cell, and it will uncoat, and then the entry of this viral nucleic acid will move into the host cell's nucleus. Once it's in the host cell's nucleus, in step 3, right, this viral genome then uses the host cell's own genetic material. It will replicate, and then in step 4, we see that it will start to assemble viral nucleic acids and proteins that belong to the virus. They're viral subunits. So then the virus will take these and construct completely new virions. That's our step 5. So ultimately, we have a release of the virions, which then leave the host cell and spread to infect other cells. So remember, this is why we use viruses for gene therapy. We've talked about that before. Most of the time, the body can fight off viral infections through cell-mediated or humoral immunity, but viruses are very difficult to kill because they live inside the cells. Any drug, therefore, that kills a virus may also kill cells. Now, most of our viral illnesses are pretty bothersome, but they're survivable. We say most because not all, right? Vaccines have been developed to prevent some of the illnesses, like polio, smallpox, chickenpox, shingles, but not all of them. And there are viruses that are long-term or chronic and lead to fatal complications. We have studied those, like HIV. Later, we'll study hepatitis. There is effective drug therapy available for a small number of viral infections. When we consider the number, it's pretty big, the number of viruses, not very many treatments or cures. So antiviral drugs are all synthetic compounds, and they work by killing or suppressing the virus, and they do this by destroying the virions or inhibiting the ability of the virus to replicate. That's how we control them with current antiviral therapy. But remember that even the best of the antiviral medications don't completely eradicate a virus. the body's immune system is still required. And what the current antiviral medications do is that they allow the body's immune system increased ability to control or eliminate a virus or limit its effects. And that's done by limiting the virus's ability to replicate or it's stopped. The immunoglobulins are these concentrated antibodies, and we've talked about those in the past. They can be nonspecific or specific to a virus. Specific ones would include things like rabies or varicella zoster. The immunoglobulins can technically be considered antiviral drugs, but we more commonly refer to them as immunizing agents. Like I said, there are relatively few viruses that can be controlled by antiviral therapy when we consider the overall number of viruses in our environment. So some of the viruses controlled by current antiviral therapy are cytomegalovirus, hepatitis viruses, herpes viruses, human immunodeficiency virus, influenza viruses or the flu, and respiratory syncytial virus or RSV. Now, the key characteristics of the antiviral drugs are that they're able to enter the cells infected with the virus, and they interfere with that viral nucleic acid synthesis or the regulation of that viral nucleic acid. So some of the drugs interfere with the ability of the virus to bind to the cells, and some of them also stimulate the body's immune system. So we approach antiviral treatment from these different aspects to try to get a larger coverage. Best responses to the antiviral drugs then are in a patient with a competent immune system. We know that a healthy immune system works synergistically with the drugs to eliminate or suppress the viral activity. Therefore, when we have a patient who's immunocompromised, he or she is at increased risk for an opportunistic infection. You may remember from the HIV lecture that opportunistic infections occur in immunocompromised patients. They would not normally harm an immunocompetent person. So they sometimes require long-term prophylaxis and anti-infective therapy. They can be things like viruses, other viruses, fungi, bacteria, protozoan. The most common examples of our immunocompromised patients, other than those with HIV, are those with cancer, those taking immunosuppressive treatments for things such as organ transplant, or those with full-blown AIDS. And the patient with AIDS is at high risk for developing these because we remember how that works, and we'll talk about that again in just a few minutes. So our antiviral drugs are classified by their nucleoside or their nucleotide activity. Your textbook has divided the antiviral drugs into two categories. They've divided them into those that treat HIV and those that treat non-HIV viral infections. Let's talk about some of those other infections. So the herpes simplex viruses, we have HSV1 and HSV2, and they cause very painful lesions that are highly transmittable through oral contact and sexual contact, respectively. There are periods of latency where there are no lesions or other symptoms. The antiviral drugs, usually acyclovir or famivir, are used to reduce the severity of the outbreak or duration and encourage remission, but they can be used prophylactically as well. Antiviral drugs, however, do not cure herpes. And if a pregnant woman has herpes, it's recommended that she deliver by C-section if there are any genital lesions present. Now, some obstetricians will prefer to deliver all infants of mothers with HSV2 via C-section before rupture of the membranes to prevent transmission of that virus to the neonate. We'll discuss this in greater detail when we get to maternal health during our third semester. The human herpes virus and varicella zoster viruses. So the chickenpox and shingles, chickenpox is HHV3, varicella zoster is our shingles. chickenpox is usually not treated with an antiviral drug unless there's some other problem or some other compromise, because it's pretty self-limiting, but it is highly contagious. It's spread by contact with weeping lesions, can also be spread through contact with droplet inhalation. Now, shingles is caused by a reactivation of the varicella zoster's virus. This most often occurs decades after the chickenpox infection. These lesions will follow along nerve tracts that are called dermatomes, and they're incredibly painful. So painful, in fact, that this is one of the times that we may see opioids prescribed, even in our geriatric population. We also will use topical application of things like capsaicin or lidocaine, which sometimes helps with the pain. We use the antivirals here to speed up the recovery and to promote remission, but they are most effective if they're started within 72 hours of symptom onset. Our Epstein-Barr virus, HHV4, is associated with mononucleosis and chronic fatigue syndrome. Our cytomegalovirus, HHV5, can cause a very serious eye infection. And then we have our HHV8, that's our Kaposi sarcoma, is an oncogene virus that actually causes a very specific cancer in patients that are HIV positive with AIDS. HHV6 and 7 are really not clinically significant. They're associated with xanthum subitum, what you might commonly know as roseola or Sixth disease or roseola infantum. And so antibodies to HHV6 and 7 are almost universally present in everyone by the age of 5. So how do these non-HIV antiviral drugs work? Well, their mechanism of action is that they inhibit viral replication. They decrease the concentration of the virus where the patient's immune system is then able to eliminate the virus. If elimination does not occur, the virus may enter a dormant state or may replicate in low levels, which can then be suppressed or controlled with antiviral therapy ongoing. Non-HIV antivirals are also, they're used to treat things like influenza viruses, HSV, VZV, CMV, and our hepatitis A, B, and C. We'll cover hepatitis in greater detail when we cover chapter 47. Our antiviral drugs, the non-HIV side effects or adverse effects vary with each drug. So healthy cells are often killed, resulting in some serious toxicities. The more serious adverse effects are listed on table 40-2 in your textbook. Please be sure to review that table. Let's talk about some of them very specifically. Amantadine or Simitrel. This is a very narrow antiviral spectrum drug. It is active only against influenza A, and the most recent guidelines don't recommend use for treatment or prevention, but that's because recommendations for the flu treatment change from year to year. It's based on this strain that's prevalent during that year. So a couple years ago, I know, for example, my daughter had influenza A. She was very sick. That was the drug that was used to treat it. With this drug, some of the CNS effects are insomnia, nervousness, lightheadedness, and then GI effects are anorexia, nausea, and some of the other diarrhea, things like that. Now, Rimonabine or Flumidine is the same spectrum of activity and mechanism of action and indications as amantadine, but it has a longer half-life. And so because of that, it has fewer CNS adverse effects. It does, however, continue to cause the GI upset. When we look at our acyclovir, this is a synthetic nucleoside analog. It's used to suppress the replication of the herpes simplex 1 and 2 and the varicella zoster's virus. It is actually the drug of choice for the treatment of both initial and recurrent episodes of these infections, and some providers will actually order this for patients in that initial state. Then they will order a refill of that for the patient to have on hand, because these drugs work very well when they're given within 72 hours of the onset of symptoms. And so once a person has the herpes virus or the varicella zoster, the shingles virus, they're familiar and they know when an outbreak is going to occur. Because